Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. In the last few classes, we have talked about identification of variants and different types of variants. So, the time has come now to talk about transcriptome analysis, and we will talk about RNA sequencing. In this class, we will introduce what is RNA sequencing, different aspects of RNA sequencing and what are its advantages over the earlier methods. Okay, so, let us start. So, we will talk about these RNA sequencing steps in brief, and we will also compare with microarray and talk about advantages of this NGS based transcriptomic analysis. Okay, so, these are the keywords we will come across: isoform, short read sequencing and long read sequencing. So, we will start with that flow chart for NGS data analysis, where you see this we have the read data from sequencer and comes in different data formats. We have data quality control and then we have read mapping, right. So, we have talked about all these steps in detail, and we have completed the genome analysis in the last few classes. So, we will talk about the transcriptome analysis now, and we will follow that up with discussion on read assembly for reference genomes, new reference genomes, and then we will talk about the epigenomic studies. Okay, so, we will start with the transcriptome analysis. So, transcriptome analysis is also called RNA sequencing or RNA-seq, even though sometimes we are not directly sequencing RNA, as we will see in some of these methods. And most of these experiments are done to identify difference in expression levels of genes between samples or between different conditions. So, it could be disease versus healthy, or it could be some sort of treatment that is given to certain samples, and you want to compare treated versus untreated samples. Okay, so, these are the main goals of transcriptome analysis, and we used to do them using microarray earlier. So, what is the protocol? How do you do this? So, some of this is actually shared with microarray protocol. So, what you have in the first step is total RNA extraction from the sample that you are working with. So, it could be, for example, the tissue sample, it could be cell samples, right. So, we do our RNA extraction, and there is something called an mRNA enrichment, because we want to measure expression level of genes, right. So, we want to get rid of other RNAs that are present in the sample, for example, ribosomal RNA, etc. So, when you isolate total RNA, you will see most of the RNA is ribosomal RNA, right. That is in the most of the RNA in the cell. So, we want to get rid of that, and we perform a step called ribosomal RNA depletion and mRNA enrichment. So, this mRNA enrichment can be done in different ways. For example, we can use something like amplification using primers against the poly A tail in case of eukaryotes, and we can amplify only the mRNA, and we can reduce the concentration of ribosomal RNA. Now, if we use this amplification, of course, we need to keep in mind that that will introduce certain biases, which we will talk about in the next few classes, the biases that we introduce during these experimental steps. So, why is it important? Because these biases will come in the data analysis, right. So, we need to deal with those when we are analyzing this kind of data from RNA-seq experiments. Okay. The better method is the RNA depletion, and this is something that people use without amplifying the mRNA, but in some cases if you have very low amount of mRNA or total RNA, you would have to perform some amplification steps that will of course affect your results. Okay, so, once we have isolated these mRNA molecules, we do something called cDNA synthesis using reverse transcriptase, and again this will introduce certain biases in our data. Okay, so, these biases we will have to deal with when you actually analyze the data. Once we have cDNA, we can proceed for this library preparation, which is adapter ligation. If you are going with Illumina platform, you have to do bridge amplification, etc., or if you are going other platform, you have to perform some platform specific steps and followed by the next generation sequencing. And if you are going for microarray, you have to prepare this cDNA for microarray experiments. It could be addition of some sort of molecule or some sort of fluorescent molecules, etc., right. So, and then hybridization in microarray. So, what is the main goal of this kind of analysis is something called differential gene expression analysis. As I mentioned, we want to compare expression levels of genes between these two different sets of samples which have been processed through the exactly same way. So, this is what we will discuss again, the differential gene expression analysis, it will come again later on, and this is a comparison of two sets of samples. So, what I will do is now we will just briefly mention how it is done with microarray or we used to do them earlier. For gene expression analysis with microarray, we can have dual channel technology or single channel technology. So, we will discuss what is dual channel technology and what is single channel technology. So, let us start with dual channel technology first. So, as I mentioned, we do differential gene expression analysis where we have two sets of samples. In dual channel technology, these two samples are labeled with two different dyes. Okay. So, you get two different fluorescent levels. So, you can imagine this, right. So, you have sample 1 from which you get this RNA molecules, then they are converted to cDNA, and you have sample 2 from which you also get RNA molecules which that are converted to cDNA according to the flow diagram. Now, once they are converted to cDNA, so this one will get one type of tag, so of one color, right. So, let us say this is color 1 and the second sample will get a tag of different color, let us call it C2, right. So, this is usually red green coloration. So, C1 is usually red, C2 is green. So, what then happens is that we mix these two samples and hybridize against the microarray chip. Okay. So, in the microarray chip, if you remember, so we have these probes already attached. So, these cDNA molecules will go there and hybridize. Now, if you have higher expression of a gene in S1, what you will see is that color 1 will dominate, right. So, if color 1 is red, you will see red color coming from this region. Okay. And if S2 expression, the expression of a gene in S2 is higher, then you will see color 2 dominate in the microarray. If these expressions are balanced, then you will see balanced color. So, you can imagine, right, if you have red green colors. So, if one sample is dominating, so you have red color. If you have S2 dominating, you will get green color, but if the expression is balanced between these two samples, then you will get yellow color. Now, in single channel technology, you do not have these two colors. So, what we do is both these samples. So, again, if you have S1 and S2, they get the same kind of level. Okay. So, the level is same and they are hybridized separately. So, there is no mixing and they are hybridized separately on two different chips, and what we do is we measure the signal that is coming from these positions, right, these different positions that are measuring expression levels of genes in these two samples. So, we get independent values and then we can compare. So, this is the bioinformatic part where we actually compare the expression levels. But there are several limitations, right. So, because microarray relies on hybridization, we need to have prior knowledge of transcriptome for oligo design, right. So, we need to design these oligos or synthesize these oligos on the chip. So, we need to know the transcriptome sequence, right. So, genome sequence and the gene sequence, mRNA sequence, etc. Right. And the second major point is that you can detect only known features and you cannot detect any novel transcript. For example, there is a novel transcript, right, or a splice variant that is expressed by in the disease sample, but since we did not know about that, we did not design any oligo to be printed on that chip. Okay. So, in that way, in that case we will not be able to identify that novel transcript in microarray experiments. The other limitation, of course, you can imagine, right, these transcript sequences are different from one organism to another, and then there are new genes, different genes in different organisms. So, you have to have organism specific microarray chips, right. So, you would find disease array, human array, etc. The other major limitation of microarray technology is the limited dynamic range. Okay. So, the expression level is detected by hybridization. So, you can have, let us say, for a gene you can have 1000 copies maybe, right, because of the space constants, right, because you want to represent all the genes that are expressed in a cell, in a sample, right. So, you want to have all of them in the chip. So, for each individual gene you can have, let us say, 1000 copies of oligos against which the cDNA can bind. Now, if the gene is expressed at much higher level than 1000, right. So, it is producing maybe, let us say, 10,000 mRNA copies, right. So, would you be able to detect that? No, right. So, because you will reach that saturation because you have these only 1000 copies of oligo. So, this means you are limited by this dynamic range, right. So, you cannot kind of go very high because you will reach saturation. You can also cannot go very low because the signal to noise ratio is low, right. So, again you will see there is a problem in detecting lowly expressed transcript, right. So, if you have only 1 or 2 transcripts of a gene, you will probably not get any signal at all, or you will not be able to detect the signal that is generated. So, you will miss the expression level of these genes that are expressed at very low level. And again, so you cannot compare whether the expression level of these genes that are expressed at a very low level, whether they have changed between your normal and the disease sample. The other limitation is, of course, is that you need a lot of RNA samples to actually do these hybridization experiments, and again in some cases they may not be available. So, because you would have to have a lot of starting material to get this large amount of RNA sample, and also because this method relies on hybridization, which could change depending on the hybridization conditions. If there are some fluctuations in the hybridization conditions, that can change the hybridization pattern, right. This could affect signal, and again this could lead to reproducibility issues. So, from one experiment to the next experiment you might not get the same results. So, that is where the gene expression analysis with NGS comes in, right. The principle is very simple when you are analyzing with NGS, is that the number of count or count of reads that map to a gene is a proxy for the number of transcripts and hence for the gene expression level. Okay. So, in microarray it is the hybridization signal, right. The number of copies that you have that will hybridize and that will generate some signal, and this signal is a good indicator of expression. In case of next generation sequencing or RNA sequencing, the number of or count of reads that we get mapping to each gene is the signal for the expression level. Okay. So, something like this if we present in a cartoon, right. You have gene A, gene B, gene C, right, and you have the control. For example, we have the normal, which is the control, and target may be the disease, and in this case you have gene A, gene B, gene C. We are comparing expression level between these two samples, and you just what you do after sequencing, you map the reads, and most of the reads are supposed to map against the genes, right, because they are coming from mRNA. And you see, for example, you compare this and you see, okay, in gene B, in the target sample you have very less number of reads and fewer reads. So, you can probably tell, okay, perhaps this gene B is showing low expression in this target sample compared to the control. Okay. And for gene C, you might see, okay, there is some increase in the number of reads that are mapping to gene C in this target sample. So, there is probably an increase in expression of gene C in this sample. Okay. So, this is a very rough idea. Of course, you cannot just look at this count and say, okay, there is change in expression. You have to have a very robust and rigorous statistical analysis that has to be done. Okay. So, and we will talk about this statistical analysis a bit later, and the tools that actually will do this for you, and we will actually do that hands on as well. We will analyze this kind of data hands on and see the differential expressions, right, identify the genes that show change in expression between two different samples. Okay. Now, when you are doing this gene expression analysis, you can do this with short read sequencing, for example, Illumina platform. You can do this in long read sequencing now with PacBio or Oxford Dadapore, and you can do direct RNA sequencing using Oxford Dadapore, right. So, if you remember the principle Dadapore platform, you can actually directly pass RNA and you can detect the signals and identify the bases. Okay. So, let us talk about the short read sequencing. What would be the protocol in this case? So, you have the total RNA extraction, you get the mRNA enrichment and cDNA synthesis, and followed by that you have adapter ligation, you have PCR amplification. This is the bridge amplification step that we need to perform, and then the sequencing. Okay. So, you have the usual steps after cDNA synthesis. Okay. And of course, you cannot sequence this with RNA, you have to synthesize cDNA. If you are going with long read sequencing such as SMRT, right, the steps are pretty much same, right. You have cDNA synthesis, adapter ligation, PCR amplification and sequencing. And if you want to go with Oxford Nanopore, you can skip the PCR amplification, right. You can directly pass the cDNA after adapter ligation through the sequencer, and the current signal, right, that will change, that will indicate the bases, right, and we can then do the mapping back to the reference sequence. The direct RNA sequencing using Oxford Nanopore, it is actually much simpler, right, and you can see you can go from total RNA extraction to mRNA enrichment and then adapter ligation and sequencing. So, this direct protocol might be quite useful in some cases, because you are introducing less number of amplification steps, and this might give us better results. So, any amplification will have some bias, right. It will amplify certain fragments, certain genomic fragments much better than other because of their sequence preferences, GC content, etc. So, all those factors come in play, and that is why having less number of steps without amplification is actually very good for our analysis. Okay. So, what we do is what we can do with this NGS is that we can do differential gene expression analysis or DGE analysis, which we will do later, and we will discuss this in much more detail, what kind of statistics we use, etc. We can do something called identification of isoforms and alternative splicing events. Of course, this was also possible in microarray experiments, but in that case you would have to design those oligos again, right. You would have to think about what kind of alternative isoforms you might see, and then design oligos against those, right, and this becomes very tedious, right. If you have to do this for, let us say, thousands of genes, this becomes a very tedious process. Whereas in NGS you can do it very easily, because this is a direct sequencing process. So, you can identify what isoforms you are getting. Okay. So, we will talk about isoforms, different isoforms that are possible. And we can also study the role of non-coding RNAs. We can quantify non-coding RNA expression levels, right, and we can study their role in expression regulation. So, let us talk about different types of isoforms that could be there in a cell. So, I have drawn some scenarios, and there could be few more. So, the first scenario here, this is the normal scenario, right. You have these 3 exons in blue, orange and green, and they are combined together to give the transcript. Okay. So, this is the normal transcript here that you get, okay, with all the exons. Now, if you have these different types of processes that can generate these isoforms, right. So, one type of process is that you can have alternate transcription initiation, right. So, transcription initiation happens at different place. Okay. So, here maybe the transcription initiation happens in this place given by this red arrow, and what that means, you will end up with only these last 2 exons. Okay. The first exon will not be there. You can have other types of cases, right. For example, you can have alternate 5' splice site, right. So, what you can have is that instead of splicing happening right at this boundary here, right at the end of this exon, it can be shifted a little bit, right, and this shift means you will include a larger region after the exon 1. Similarly, you can have alternate 3' splice site, right. So, splicing happens slightly upstream compared to the original splice site, right, and then again you include a bit more region with exon 2. And finally, this is the classic case, right. So, you can have a skipped exon, right. So, maybe the second exon is not included, only the first and the third exon they combine together and then they give rise to this transcript. You can have other kinds of scenarios again which you can find in the reference. The advantages of short read sequencing, right, because we know already Illumina is really high throughput, you get a more large number of reads quite easily and not very expensive. And we also know the biases and error profiles of Illumina method, right, and we have an established data processing pipeline. We have already seen, right, when you are doing this variant analysis, we have already seen some of these pipelines that are available, and most of them are developed for FASTQ files, right, for Illumina platform. What are the drawbacks then, right? So, why don't we always use Illumina, or why do we need the long read sequencing at all? So, one of the first step is the multi-step sample preparation, right. So, we have seen for short read sequencing we need this multi-step preparation, we need this bridge amplification step, right, and this can introduce bias in the data. Okay. So, this is a problem. Then you also have limited isoform detection, right. So, you can imagine, right, if you have a long gene with multiple exons and you are working with short read data, even with paired end data you will not be able to identify all the isoforms if they are present, right. For example, in case you have, let us say, alternative initiation sites, alternative splice sites, they may not be detected in the short read data. And in case you want to discover these isoforms, right, so you may have to do this 3 to 4 transcript assembly for this transcript discovery. Again, any assembly process, right, it becomes computationally more challenging. So, here comes the long read sequencing, right. So, that can probably address some of these issues of short read sequencing. So, long read sequencing, it can sequence full length transcripts, right. So, we do not have to worry about this assembly of short reads to get the full transcript sequence. So, in long read you can sequence the full length, and which means the computational analysis is simpler, you do not have to again go through all this transcript assembly process. And it can also discover these isoforms, right, because it is long read. So, if it is like 1 kb, 2 kb or even 5 kb sequences you are getting, you will probably discover most of the isoforms that are out there, right. You do not have to worry about again going to assembly and then trying to find the isoforms. Okay. So, this is actually a big advantage of long read sequencing. What are the drawbacks then? So, one is of course the low throughput. So, they have lower throughput than the Illumina platform at this point, but of course, this might increase in future. And also you have multi-step sample preparation in this process also, right. If you are going for packed bio SMRT, you have to do the amplification again, and this can introduce bias. So, this is where the long read direct RNA sequencing comes. Okay. So, it retains the advantages of the long read sequencing and also addresses some of the drawbacks. Okay. So, as we have seen, it can sequence full length transcripts, and computational analysis is very simple. It can discover these isoforms. In addition, it can detect some of the RNA modifications. Okay. Because it is directly sequencing RNA, any chemical modification on the RNA that will change the current signal. Okay. And so, this information can also be collected during RNA sequencing. Okay. So, this is the advantage of direct sequencing, and it can also detect the poly A length because it is directly sequencing the RNA. So, it is not through any amplification step, so which can change or which can make this estimation process very difficult. So, what are the drawbacks then? Why do not we do direct RNA sequencing? So, again the throughput is still lower compared to the Illumina platform, right, or short read platforms, and in some cases this might be a limiting factor because you can you want expression levels of even the very lowly expressed genes to be measured, and in that case you need higher amount of data or bigger amount of data, right. So, you need a lot more throughput. So, at least 30, 40, 50 million reads to actually get to that level, right, where you can measure expression levels of even very lowly expressed genes. Okay. So, in that case you probably want to use something like a combination of long read and short read sequencing, right. So, because of this, because then you can combine the advantages of both of these methods. Another drawback of direct RNA sequencing is that we still do not understand these biases and errors very well, right, because we have been doing this method for a very short time, and more time is required to understand what kind of errors you can get and how to address them. Now, there is something called strand specific RNA sequencing. We have talked about RNA sequencing in general up to this point. So, there is something called strand specific RNA sequencing. So, why do we need this strand specific RNA sequencing? Because we have not talked about the strand specific genome sequencing, right, because in that case it was not really required. So, in case of RNA sequencing, you can imagine a scenario, right. So, you have the genome and two genes are on two different strands, but they still overlap. Okay. And in that case, right, so you have these two genes on opposite strands, they can overlap with each other, right, and they will produce mRNA that will be processed, and then you are doing the RNA sequencing. And after the RNA sequencing, you get the data, you get some read, and that maps to these overlapping regions. Now, which of these two genes is producing these read? Okay. So, this is something that is very difficult to figure out, right. If you have this kind of overlapping genes in two different strands in a genome, you cannot tell, right, which gene is producing this mRNA, and you are getting the read data from that expression of that gene, right. So, this is a challenge, right. So, you can if you have reads mapping to the overlapping regions between two genes, the read can come from either of them, right, and we cannot determine this with normal RNA sequencing, and this is where you can actually do the strand specific or stranded RNA sequencing, and it can remove this ambiguity, and it can measure these expression levels of these genes much more accurately, right, because you can now resolve these reads much better. So, how does this work? How do you do this strand specific RNA sequencing? So, what we do is we add these adapters 3 prime and 5 prime adapters sequentially, right, and these adapters are unique for 3 prime and 5 prime, and we can add them in a sequential manner with purification steps in between. Okay. So, that now once we get an adapted sequence, we know, okay, we are getting the forward strand or we are getting the reverse strand data, right. So, this helps us in separating out this strand information from the read data. Okay. So, here is the brief schematic, right. So, we have the mRNA, and this is added with 3 prime adapter in orange, and then we do ligation and gel size selection. So, you might ask why we did not get attached to the other side, right. There are ways to actually make sure that this will only attach to one side, okay, and this side can be prepared for that way. And then you have the 5 prime adapter added on this side. Again, you can specify, right, you can control this, regulate this process, which side where it will be added, and you do the final product with these 3 prime and 5 prime adapters, and these are unique ones. Now, once you sequence, right, go through all the processes and then sequence, you will get the adapted sequence, and you know which strand you are getting the sequence from, right. Based on that, you can assign which gene is producing this mRNA and the read data, right. Okay. So, the one more thing that comes in RNA sequencing is something called small RNA sequencing. Okay. So, this actually is a part of the transcriptome, right. So, you have normal mRNA, but also you have small RNA molecules inside cells. So, this is something we know now, right, and this is the small RNA sequencing means sequencing of small non-coding RNA molecules such as micro RNA. Now, so far we have talked about sequencing of mRNA, right, mRNA enrichment, etc., ribosomal RNA depletion, etc. We have not talked about this micro RNA. So, how do you go about it? So, the first step is same, right. So, we have the total RNA extraction from samples, and in this, after this step, what we do is we then do something called size selection, right, for small RNAs only, right. So, we can separate out only the small RNAs that are of certain size, right. You can say, okay, we will separate out maybe only the 35 base pair, up to 35 base pair RNA molecules or up to 50 base pair RNA molecules, and you can separate out these molecules and do the RNA sequencing. Okay. And once you do the RNA sequencing, you get the expression level of these microRNA or these non-coding RNA molecules. So, this information is very useful, because we know now these molecules are involved in expression regulation. So, knowing their expression would be very useful in understanding what is happening inside the cell. So, we can now move into the advantages of RNA sequencing after all this discussion. So, one of the first thing we have seen is that it has the ability to detect novel transcripts and isoforms quite easily. We can combine short read and long read sequencing to actually identify all these variants very easily. The second is we have a very big dynamic range compared to microarray, right, because we are not limited by number of oligos that are available for binding or hybridization, right. We can sequence, if you have 10,000 copies, we will sequence the sequence data will be proportional to that, right. So, this will give us this flexibility, right, if from let us say very low expression to very high expression, and this means we can detect this rare and low abundance transcripts quite easily. Now, coming to the challenges, of course, it is not perfect, right. There are a lot of challenges when you are dealing with this RNA sequencing data, and some of them can be addressed when we are doing the bioinformatic analysis, right. So, there are biases in the data, right. So, we have mentioned some of this. You have something called amplification bias, you have the selection bias, right. So, all these things come into play. This is something we will discuss in much more detail in the next few classes, and we will talk about the methods that can address some of these biases. It can be expensive and time consuming RNA sequencing, because you have to sequence a lot of samples to quite a bit of depth, right. As I mentioned, 30-40 million reads or even more. If you want to identify these very lowly expressed genes. So, this could be expensive, and preparation of the library could be time consuming. One of the things that RNA sequencing still does not do very well is the transcription start site mapping, right. Because of these amplification steps or even sometimes direct RNA sequencing, you do not get information about this transcription start site, okay, and this is something that can also be now addressed by devising different ways of protocols. Okay, here are the references that we have used for this class, and to summarize, we have talked about transcriptome analysis through RNA sequencing, and we have compared against microarray. We have seen there are several advantages of RNA sequencing, and that is why we do RNA sequencing these days more regularly than microarray. We talked about the short read, long read and direct RNA sequencing. We talked about the advantages of each of these methods as well as the drawbacks, right, and perhaps we can combine these methods short read, long read or short read, direct RNA to address some of the limitations that each of these methods have. So, then finally, we can use RNA sequencing to detect isoforms. So, we talked about different types of isoforms that are there and novel transcripts, and finally, we can also do small RNA sequencing that can help us understand this role of small RNA molecules in gene expression regulation. Thank you.